Good evening, everyone. Welcome to our Science in the News talk tonight. My name is Peter Yang, and I'm a second-year PA student at the Yale School of Medicine. I'm joined today by three of our amazing speakers today who are from the graduate school here. For those of you who've seen one of our talks before, welcome back. And for those who are new, some quick background information about Yale Science Communication. We're a graduate student organization that aims to ignite scientific engagement across diverse communities and train effective science communicators. So each year we speak at various venues from libraries to local bars across New Haven and beyond about science. And we believe that anyone can engage with science and use it in their lives. So it's our responsibility to make it accessible to the general public, especially underserved communities. And we're really glad that you could join us today. So before we begin, I'm going to introduce the speakers that we have. So first up is Tayah. She is a fifth-year graduate student in the chemistry department, and she studies the gut microbiome, which is the bacteria that live in our gastrointestinal tract. She's from Wadsworth, Ohio, and a fun fact is that she's a certified spin instructor who's coached over 400 classes. Next, we have Isabel or Isa. She is a second-year PhD student in the astronomy department, and she studies a phenomenon called fast radio bursts and how they help in galaxy formation. She's from Philadelphia, Pennsylvania, and her fun fact is she's an avid baker, and her favorite pastry to make is peach pie. And last but certainly not least, we have Anjana, who joins us from Kerala, India. She is a third-year PhD student in molecular biophysics and biochemistry, and she studies proteins in squids, specifically how squids put them together. And her fun fact is that she's a polyglot who can speak 6 different languages. All right, so sorry about that. Before I continue, I just wanted to say, each of our speakers will be giving their talks in sequence, so we'll save all of your questions for the end. If you have anything that you think of that you want to ask, please write them down or file them away in your mind, and then we'll take all the questions at the end. All right, so to give everyone a little bit more background about our talk, as you've seen, right, our title is Species Spotlight. So each of our 3 speakers today is highlighting a particular animal species and the adaptations that it has to really allow it to do what it does the best, right? So if you're anything like me, when I was growing up, I was, really fascinated by animals. And, obviously I'm not in this picture, but I think as kids, we all really do have a pretty deep fascination for the natural world. I know when I was in like middle school and elementary school, when we'd have like rainy days and be watching movies in school instead of class, I would always love when my teachers would put on like an Earth documentary or something else fun like that. So if you've ever watched one of these, I'm sure a lot of you have thought, how do these animals do that? Or how do they manage to pull off what they do? So today, that's one of our main themes is we hope to highlight for you some of the special ways that animals have really evolved and adapted to fit into their environments. So to that end, we have 3 species here for you, 3 stars of our show, if you will. So first, in our Deep Freeze section, Tayah will be taking us back in time to learn all about the woolly mammoth and how it might not actually be a thing of the past. Then second, we have our Arctic Anthem section, where Isa will dive into the physics behind how beluga whales make whistles, clicks, and other vocalizations that help them survive in the frigid oceans. And then thirdly, Anjana will teach us all about the incredible camouflaging capabilities of squids in her Shining Seas talk. And if any of you have seen the brief introductory video that we sent out, you may know that we have a little bit of a competition going on. Each of our speakers wants to convince you that their species is the best. So as you listen to the talks, we encourage you to think about which one of them is your favorite. And in addition to spotlighting each of the adaptations and the unique species tonight, we also just wanted to remind everyone that, in the busy, hectic modern society we live in, sometimes it can be hard to find time to enjoy nature and appreciate animals, plants, things like that. So we hope to remind you all that, we're all connected. And we wanted to share this quote from Aristotle with you all. In all things of nature, there is something of the marvelous. So with that, I will turn it over to Tayah to kick off the first portion of our talk, where she'll teach us all about the elephant in the room.
Great. So thank you, Peter, for that awesome introduction. And I am so excited today to be kicking off our species spotlight. My name is Tayah, and again, I'm going to be introducing you to a topic that blends my past love of ancient life with my current scientific excitement. So ever since I was a little girl, I have loved dinosaurs. Hopefully some of you can relate. And even when I was in kindergarten, I told everyone that I wanted to be a paleontologist when I grew up. Now, life leads us down many different paths. And although I haven't been able to, become that paleontologist and have been led slightly astray, I have still been able to maintain my passion for dinosaurs and really appreciate them from a new lens. Now, one of the first things that you may think of when you hear dinosaurs is Jurassic Park. In case you don't know, Jurassic Park is a series of movies where scientists are able to bring dinosaurs back to life through science, and they do this on a series of islands where people can then visit them to see what it was like when they were around in the past. Now, this isn't only a topic that is in movies, but this is also seen in museums. So think about the last time that you went to a natural history museum. It's very common for these lobbies to open up to large displays of dinosaurs and skeletons so that people can really picture what it would have been like to walk amongst these ancient creatures. And while this is seen on the big screen and in your museums of things of the past, I want you to think today of, is this something that could be brought to the future? Can we bring extinct creatures back to life? Now, in Jurassic Park, dinosaurs are brought back to life through the discovery of a mosquito with blood in its abdomen, and there was dinosaur DNA in that mosquito. Now, scientists actually have been able to find mosquitoes with blood in their abdomens from ancient times, but there's no DNA that remains. And this is because DNA over time fragments and degrades. Now, a little recap. DNA, which is shown here, contains all the information responsible for an organism's development and function. And while we're talking about science, let's go into a little bit further of a scientific recap to help with the rest of the talk, as well as some of the talks you'll hear later tonight. So again, first reminder about DNA. It's shown here, this double spiral structure, and it contains all the information responsible for our function and development. Now, scientists are able to use DNA sequencing to look deeper into the DNA and how it's made up. So using DNA sequencing, scientists are able to determine the individual puzzle pieces of the DNA. And when you put those puzzle pieces together, it can reveal an organism's specific characteristics. Now, where is our DNA? Our DNA is in our cells, and cells are tiny units of life that make up living organisms. And in each cell is a nucleus, which is home to all of our DNA. Now, back to where we were. While DNA could easily be obtained from you or me, from our nails or our hair or our skin, it's much different for ancient DNA. So ancient DNA, as I said, is often very degraded and fragmented from the long time that it's been there, and it's often found in very tiny amounts. Further, it's often contaminated with bacteria or fungus. So while our DNA, if we looked at it right now, may look something like this, ancient DNA would probably look a little something more like this. There is one condition, though, that is great for preserving ancient DNA, especially in hair, and this is permafrost. Permafrost is a thick layer of soil that stays frozen year-round, and it's found in Greenland, Siberia, and Canada. Now, taking all of this into account, you may be thinking that there seems to be a better candidate for this Jurassic Park idea than dinosaurs. And that is the king of the deep freeze, the woolly mammoth, which reigned in areas that are now heavily covered in permafrost. Now, woolly mammoths, if you couldn't tell by looking at them, are related to today's elephants. Specifically, the Asian elephant is its closest living relative. A little bit more about mammoths. Mammoths were about 13 feet tall, weighed about 6 tons, but could be much larger, had thick brown fur, and large curved tusks, which could have been used for foraging or for fighting. And they were covered in this thick fur and hair, with some of their hairs even being 3 feet long. And we think of mammoths being around mostly in the Arctic, but we have to remember it was a little bit different when they were around. So they actually ranged in this entire yellow region of the map, including where we are today, with most of them dying out roughly 10,000 years ago. While you may imagine discovering a mammoth in permafrost to look a little bit magical, like this image here, unfortunately, it generally doesn't look this beautiful. But lucky for us and for scientists, some of the best preserved mammoth remains have been found over the last 15 years. In 2007, Luaba was found, who is a mammoth calf shown here. And then just 5 years later, in 2012, Yucca was found, who's a mammoth juvenile that really challenged Luaba as one of the best preserved mammoth samples. And if you looked at the news at all over the summer, you may have seen news of another mammoth that was found, Nunchuga. And Nunchuga was actually found in Canadian permafrost, which is exciting as that labels her as the best preserved North American mammoth sample that's ever been found. And so with the discovery of these permafrost samples, scientists have been able to use DNA sequencing to learn more fun facts about mammoths of our past. Now, I told you a little bit about DNA sequencing, but let's get a little deeper into how this is actually done. So first, DNA is taken from mammoth permafrost samples, and then DNA is also taken from Asian elephants, which we talked about. They are the closest living relatives. From there, scientists are able to sequence the DNA, and they're able to look at the individual building blocks and see which building blocks form genes. Genes are sections of DNA that contain information for specific characteristics. So for example, me and you both have genes for our eye color. Blue eyes, brown eyes, whatever it is, there's a gene corresponding to that. Same thing for elephants and mammoths. They have genes possibly corresponding, to their trunk. And so from there, scientists are able to compare the genes that they see in the mammoth samples to the genes from the elephant samples. Since they're very related, oftentimes a lot of these genes will be the same. As I just mentioned, they probably have very similar genes for their trunks. Another thing that they may see is similar genes that are slightly different, where mammoths would need to have genes for cold regulation, since they were around during colder climates, and Asian elephants would need to be more adapted to a hotter climate. And then probably the most exciting for scientists are the genes that are unique to the woolly mammoth, which are exciting for scientists to dig into and try to figure out what those genes did for the mammoth when they were around. Now, many of these differences that they've observed between the elephant and the mammoth DNA is due to their cold adaptations. So as I mentioned, mammoths were really the kings and queens of the deep freeze, and their DNA shows it. So some cold adaptations that scientists have found have been their large fat layers and their large curved tusks. And then some others have been genes corresponding to their small tail and ears, which are really important for minimizing heat loss. That's why they're smaller than as you would see in an elephant. Now you may say, yes, I've seen a picture of a mammoth before, and I probably could have pointed out those differences to you. But what's really fun about DNA sequencing is it allows scientists to see underneath the surface, to see really how these mammoths are the kings and queens of the deep freeze with their hidden cold adaptations. For example, scientists have found 8 genes related to internal clocks. So when mammoths knew whether to be awake or asleep, which was really important because when mammoths were around, they had to be with large periods of summer sunlight and almost consistent darkness in the winters. And they also have found other genes linked to temperature sensor alterations, meaning that mammoths were altogether less sensitive to the extreme cold. Now, I didn't really come here today just to tell you about mammoths of the past, but I came here to talk about if mammoths could be a thing of the future. And so this is the idea of de-extinction. So de-extinction is defined as the recreation of an extinct species. And while this is possible in multiple different ways and speculated, the most probable way that this would be accomplished is shown here, in which DNA would be obtained from a permafrost mammoth sample. And as we know, it's probably going to be a little bit fragmented from its time in the ice. And so DNA from an Asian elephant would be used to fill in those gaps. From there, all of the DNA from the mammoth with those gaps filled in would be inserted into an empty egg, grown by an elephant mother, and then delivered to give us our de-extinct mammoth. Now, I've talked to you throughout this talk so far about how mammoth DNA has been well preserved enough to make scientific discoveries, but now we're kind of switching gears, and are mammoths, well, the best candidate for de-extinction? So let's talk about that real quick. I'm going to have a competition within our species spotlight competition. So let's look at our contenders. We have dinosaurs, which we've talked about from Jurassic Park, mammoths, and then another option is something with a more relatively recent extinction timeframe, such as the passenger pigeon, a species of pigeon that was used for delivering letters and mail that went extinct roughly 100 years ago. So we have our candidates. So now let's think about the important factors that would be necessary for a de-extinction candidate to help us kind of place them on the podium. So number one that we've spent a lot of time talking about today is well-preserved DNA. So this is kind of ruling out dinosaurs as they were around, millions and millions of years ago. And they weren't around in areas that are heavily covered in permafrost, meaning that their DNA has not been that well preserved. Another important factor is the presence of a living relative. This is important so that you can make a lot of those DNA comparisons. Another importance is a relatively recent extinction timeframe, and this kind of dually plays into, that means generally the DNA is better preserved and there's a higher chance of a living relative. So at this point, mammoths and pigeons are both looking like really great candidates. However, there's one last factor that is very important as well, and this is the fact that mammals are a lot better candidates because scientists have way more trouble accessing the DNA in eggs. So putting our pigeons at number 2 and our mammoths with a living relative and relatively recent extinction timeframe at the top. So of course, I've hopefully sold you a little bit on how the mammoth is the best candidate for de-extinction, but what you're probably wondering at this point is this possible? Which is a great question. So currently, there are teams working to create a way for more and more scientists to study mammoth cells in the lab, and they're doing this by modifying the cells from elephants to convert them to be more like mammoth cells. Further, in 2019, scientists were able to show that mammoth cells were actually still relatively active. So I'm going to walk you through that summary of that study. So in this study from 2019, scientists were able to take cells actually from Yucca, one of the mammoths I showed you a while back now, and they were able to take that DNA, put it in an empty mouse cell, and just see how active it was, what it did. And so some of their findings here, they were able to show that the mammoth DNA and cell was able to use nutrients from the surroundings. They were able to grow, so they were able to form almost a new nucleus. And then they were also able to kind of take steps toward continuing life. So generally cells, they grow and they use nutrients and then they divide. And in that division, they split apart and then they continue to grow and repopulate. And these cells from the mammoth were able to almost split apart, but not completely. So this study really highlights how fantastic it is that this ancient DNA is still active and able to use nutrients and grow, but it also shows kind of how far we still have to go before de-extinction is really on the table. But this advancing scientific technology is really helping scientists get closer than we've ever been before. And so just kind of to sum up my argument of my species spotlight, mammoth DNA is really well preserved due to its presence in permafrost. And it's also been great for scientists because mammoths have this living relative in the Asian elephant, which they can use to compare the DNA and make really cool scientific findings, especially about their cold adaptations, which I talked about. Scientists have also been able to show not only about their past, but that they are still biologically active. And so I want to kind of leave you still with the question of, do you think mammoths are just a thing of the past and kind of kings of the deep freeze due to their cold adaptations? Or do you think they may also be a good candidate for de-extinction, making them a candidate for the future? And so I want to leave you again with some inspiration to always dream big, no matter where your life takes you, and to really look back on your past loves from your childhood, as generally you can look at them through a different lens and different appreciation. I can't wait to take your questions when we get to the end of our species spotlight. Of course, there's many that come up with the idea of de-extinction, which I can't get into in 15 minutes. But yes, thank you so much. You've heard about my contender now, but we will be moving on from deep freeze to the shining seas with Isa's Arctic anthems. Thank you again.
Hi, everyone. I'm Isa, and I am a second-year PhD student in astronomy at Yale. So usually my research focuses on a mysterious astronomical phenomenon called fast radio bursts. But today, instead of that, I'll be talking to you about my favorite animal, the beluga whale. So my talk is titled Canaries of the Sea, Beluga Whale Vocalizations. So specifically, I'll be going through the sounds belugas make, how they make them, and how they use them, and why it's so interesting. As you may have guessed from the title of the talk, we know that belugas are vocalists. But over the course of this talk, I also hope to prove to you that belugas are excellent physicists. To start, I'm going to give you a few facts about belugas that will become relevant. First, the scientific name of the beluga is Delphinapterus leucus. This translates to white dolphin without a fin. Belugas are cetaceans, or they're tooth cetaceans. Other cetaceans include other whales, dolphins, porpoises, and their closest relative is the narwhal. Belugas live in Arctic and subarctic oceans. These regions are shown here on the map, and they're carnivores that hunt fish, squids, crustaceans, worms, and other animals. So putting that information together, that brings up one major question. How does a beluga who's living in murky water find a fish that's tiny, far away, and hard to see? And how do these belugas living in this Arctic water find holes in the ice sheet so they can breathe? And the answer to that is through sound, specifically echolocation. So, as the talk title alludes to, belugas are known as the canaries of the sea. And this is because they make many vocalizations frequently, including echolocation. They can make so many different types of sounds, and they can even mimic humans. So here's a quick example of some of the sounds belugas make. The functions of these sounds range from social communication, navigation, specifically navigating under ice sheets and finding holes from which they can surface and breathe, and finding food. So to understand how belugas are overcoming the challenges of their environment, I am going to highlight some of the specific adaptations and features of their anatomy, starting with their white skin. Belugas are entirely white in adulthood, so they're sometimes called white whales. As babies, though, they're gray or brownish in color. So in winter months, their skin thickens, but they molt annually in the summer months by rubbing their skin against different objects to rub it off. And then, like their scientific name applies, belugas do not have a dorsal fin. This allows them to swim under shallow waters, in shallow waters under ice. Belugas also have very wide flippers, and they are some of the only whales that can swim backwards. Belugas, like other whales, have a blowhole through which they breathe and produce sound. And around their neck, belugas have muscles that allow them to move and turn their head. Other whales have fused vertebrae, which means they're not able to turn their heads, but belugas do not have fused vertebrae, so they're the only whales who can turn their heads, nod their heads. And then one of the most distinctive features of a beluga is its melon, which is the bulbous part of its head that you may notice when you see a beluga. And this melon is malleable, soft, and this is what allows belugas to use echolocation. So contrary to what you may expect, belugas do not have vocal cords, so they're not producing any of their sounds through their mouth. It's produced through the blowhole. To be able to focus those sounds, belugas manipulate their melon, which is malleable because it's filled with oil, so it's squishy. They can change the shape and focus the air, and it goes through sacs in their head and comes out of the blowhole. So belugas can use this to make communication with other belugas, whistles, and pulses, but they also can use this to make echolocation clicks, which are short, like clicks of a signal. It sounds something like this, and it would look something like this. So it would sound like this, and a beluga would produce a pulse of sound in a certain direction. It would bounce off something, let's say a fish, and the echoes would return to the beluga, and the beluga would know that there's a fish nearby. And I think it's very important that we understand how this actually works. So to that end, I'm going to talk a little bit about sound and how sound works. So what is sound? A sound is a vibration that travels as a compression wave through some sort of space. So for us, that space is air. For belugas, that space is water. And you can think of the sound wave as a slinky. So let's take a look at this animation. If you take a slinky, you can see that there are some regions, if you move it back and forth, where the springs are tighter together and regions where the springs are further apart. This is kind of what happens in a sound wave. Instead of springs, you'll have atoms in the air, and in some regions, those atoms will be squished together, and in between those regions, there will be less atoms in that same amount of space. They'll be stretched further apart. And this forms a wave where these regions where the atoms are squished together form the peaks of the wave, and the regions where the atoms are stretched further apart form these valleys. And these peaks and valleys move at a constant speed. So you can see these peaks of these squished atoms are moving forward. There are a few important physical laws we need to go over to understand echolocation. First is that sound waves reflect off of surfaces. So you can see this in this animation, a sound wave is reflecting off a wall. If you think about going into a cave or a small room and you make a sound, you may hear the echoes. Another key point is that sound waves get weaker as they travel. If you think about this in terms of being in a room with someone else, if someone is right next to you and they speak, it's going to sound a lot louder than if you're on the other side of the room. And a third important thing to note is that the speed that sound travels depends on the density of the space that it's traveling through. So sound travels faster through a more dense or solid medium, such as a solid like metal, versus through a more sparse medium or space like air. So this animation, this video illustrates how much faster sound travels through in a solid compared with water and then in gas. So we can put all of this information together to go back to echolocation. So echolocation is defined as the use of echoes or reflected sounds to find an object or navigate. So an animal will produce a sound and they'll get information about the location and distance of objects using both the volume of the returning echoes as well as the time it takes the echoes to reach each of their ears. So we can take a look at this equation to further clarify that. So the distance of an object is equal to the distance of an object is equal to half of the time it takes for the echo to return times the speed of sound. Because if you think about it, the echo is traveling along the way, the sound is traveling as a wave towards the object, and that takes, let's say, 10 seconds. It bounces off and then it returns, and that takes another 10 seconds. So we can say it takes a total of 20 seconds for that echo to return. And so we need to take half of that time. And to get a distance, we can take the speed. So let's take a quick pause to think about something. The speed of sound in air is 767 miles per hour. So can anyone guess what the speed of sound in water is? We know that it should be faster because it's a denser medium, but I don't think anyone can guess off the top of their head what the speed of sound in water is. And I can't either. It's 3,315 miles per hour. But if you think about what we just talked about in terms of echolocation, we know that belugas innately know the speed of sound in water. To be able to use echolocation to find objects, they must know this number in their head to perform those calculations. And more than just getting locations of objects through echolocation, belugas are able to get density of objects, the speed at which those objects are traveling through calculations involving echolocation. And this all happens almost instantaneously, and they themselves are moving through water when they do this. So that's pretty incredible. And I can also take this moment to relate this back to my own research. So as I mentioned in my introduction, I'm an astrophysicist and I study something called fast radio bursts. Fast radio bursts are light waves. And what we want to do with them is be able to measure the objects between us and where these fast radio bursts are coming from. So it's a very similar problem to what the belugas are doing, instead using space as the medium and a light wave. So it's pretty incredible that belugas are already kind of showing us a way we could do our own research. And in the last part of this talk, I'm going to go over a few facts about beluga conservation. So currently there are about 150,000 belugas in the wild, and they're classified as near threatened but not endangered, except for a few subpopulations. And one such subpopulation is the Cook Inlet, Alaska population. There are now under 300 belugas in that pod, and their population is mysteriously declining every year pretty rapidly. So belugas are facing many threats, and one of those is ocean noise, which is caused by humans. And this is a big problem because, as we talked about throughout the talk, belugas solve the challenges of living in the Arctic by using noise and echolocation to find food and places to breathe. And this is how they navigate and communicate with each other. So ocean noise pollution is a very big deal and is very negatively impacting their populations. Scientists in the past have been able to show that belugas can adapt to increasing noise levels, but they've also studied baby beluga communication with their mothers, and they found that baby belugas cannot adapt to the increasing noise levels. So With noise pollution, baby beluga calls to their mothers are getting masked. But this is also an opportunity for us to study belugas. So going back to the Cook Inlet, Alaska population of belugas, scientists have noticed that this population is very rapidly declining, but they've thought of a key way to be able to track the population and try to understand what's causing the decline. And that is by tracking their echolocation clicks to understand the relationship between beluga presence and feeding and the presence of fish and other sources of food. So by doing this, they're hoping to be able to understand why this population of belugas is declining so quickly. So in summary, belugas are the most vocal of the whales, and so they're called the canaries of the sea. And they use their vocalization to solve many challenges of their environment, but complex physics underlies this. And finally, belugas face many threats, especially ocean noise pollution, but we can use their vocalizations to learn how to help belugas. And I'm going to end with this little video of a baby beluga. Oh. Okay, it's not working, but I think I hope I've convinced you that belugas are pretty amazing physicists. What they do is pretty incredible. And now we're going to go on to Anjana, and she's going to talk about an animal that uses another type of wave, light. Thank you.
Hello, everyone. I hope you can hear me. My name is Anjana, and I'm here to talk about the art of coloration in squids. So before I start, let me tell you, belugas are not the only one that took a course in advanced wave physics. I have here the star of my show, squids, who were right there as their classmates. So let's dive right in. Light, as you know it, that surrounds us is made up of a rainbow, like all the colors in the rainbow. And when that light that is composed of all these colors comes in contact with an object, it can either reflect like in a mirror, or it can pass through or transmit like through glass, or it can get completely absorbed where, like in a dark room where you see no light. So keeping these properties of light in mind, when you see an object, let it be anything, let it be an animal, let it be a plant, or any object around you, what you see as the color of the object is essentially the color that bounces off the object. That is, when the sunlight that is composed of all colors hits the surface, or let's say in this picture diagram, a leaf, the leaf has a specific chemical that can absorb all colors of the light except the color that you see. And therefore, when you say a leaf is green, what you're seeing is the green color being reflected off the surface of the leaf. So when I say most of the biological color in animals comes from a pigment or a chemical that is present on their skin, that essentially means there is a pigment that absorbs all colors of light except for the color that you see. So lion, for example, when I say lion, there is melanin, a pigment in their skin that makes its skin brown. Whereas when I say cardinal is red, that means there is a pigment that gives the cardinal feathers a bright red color. So we would think that's how all colors work, but that is not true. Apart from biological color that comes from pigments or chemicals, there is also another spectrum of color known as structural color. If you look at all the animals in this slide, you can see that they're not just colorful. They have a shiny metallic-y texture to it, right? So that actually comes from light interacting with structures that are present in these animals that can sort of manipulate light to give it an extra brightness boost to make it a little bit extra shiny, a little bit extra metallic. And I'm going to explain how structural color works using the star of my show, squid, and as you can see, they're pretty good at it. So before I jump into how squid color works, let's kind of understand the physics. Believe it or not, squids actually use the same principles as that of a colorful soap bubble to camouflage in their open ocean where they live. So think of light as a wave. If you have ever looked into a lake, you can actually see fish swimming around and whatnot. But when you shift your focus, you can actually see your reflection in water. That is because when light as a wave hits a transparent or semi-transparent surface, some of the light will reflect and some of it will pass through. Now, how does that help with a soap bubble to become so colorful? A soap bubble is essentially a layer of water sandwiched between two soap layers. When light hits the first surface, some light will reflect and some of it will go through. But when some of the light goes through, the light that passes through can actually reflect off the second surface, and the two waves can actually now interfere. When they are in phase, we are going to see them add up, and that is called constructive interference. And when they are not in phase, that is, their crust aligned with their troughs, they are essentially going to cancel out, and we are going to see destructive interference. Okay, that is a lot of physics. How does a soap bubble become so colorful though? It is because the sunlight that is surrounding us is made of all colors of the rainbow, and each color has a different wavelength. And what color actually gets amplified from the surface of the soap bubble actually depends on the thickness of the bubble. That is, when a wave of a certain wavelength hits the bubble surface, its thickness will dictate whether you're going to see that color on the surface or not. And this process is known as iridescence. What I mean by that is, here you can see a soap bubble where part of its surface is blue because the blue wave is getting amplified on that specific surface due to its specific thickness that supports the amplification of that color, whereas green gets cancelled out because the thickness of soap bubble around that region does not support the amplification of that color, therefore it cancels out. So the big take home from this slide is the thickness or the spacing between the soap layer that sandwiches water is quite important to understand which color gets amplified. And squids somehow learn this entire physics on their own in order to camouflage. Squids actually live in the open ocean where there is nothing much for them to hide behind or blend into. All they can do is sort of make themselves look transparent in order to become invisible in the eyes of a predator. Or if they want to communicate with each other, they can only change colors on their body. So let's take a closer look. Squids have mantle or a pocket of skin that covers their body. And when you take a microscope and go in, you can see there is a layer of pigment-filled cells. Pigments, as we spoke, like I mentioned earlier, can absorb most parts of light and reflect or bounce away some. And those pigments, those colors that reflects off the surface is what we perceive as the color. But right underneath that pigment tissue, we have a layer of camouflaging cells called iridophores that can actually manipulate light based on the principles that I mentioned earlier, same as that of the soap bubble. And when I take a closer look at these iridophores, they're actually made up of neatly stacked protein sheets which are equally spaced. And when I say protein here, it's not the same protein powder that we think of, or the one that we, or the meat or anything that we actually come in contact with. These are proteins that the squid makes all by itself in order to camouflage. So these are very specific proteins that the squid makes in order to camouflage. Now, I said squids can use these protein sheets to camouflage. So let's see how that works. When the sunlight that is composed of all colors hits the squid's skin, the iridophores get activated. And as I said, these iridophores are made up of proteins that are equally spaced and neatly packed. So when light with all colors hits these surfaces, only some colors can get amplified and everything else cancels out, just like in the soap bubble. And it all comes down to the spacing between these protein sheets. In this particular case, the squid is blue, which means the spacing is such that it will only amplify or give a brightness boost to blue color, and every other color in the rainbow will cancel out. But if you take a closer look at this iridophore, you can see that the color is actually changing from bright yellow to deep blue in a matter of seconds. But as far as I've explained, they were just supposed to stay blue. Why are they changing color from bright yellow to deep blue? And it's all happening within a matter of seconds. So how did these squids figure that out? This color change happens actually because the spacing between the protein sheets are changing on the squid's command. And as the spacing between these protein sheets change, since thickness is what dictates the color that you see ultimately, the spacing changes and you end up seeing a different color. So when the spacing is way farther apart, you end up seeing colors that are closer to the red region of the rainbow. And when these protein sheets are tightly packed all together, you see colors that are closer to the blue part of the rainbow. Ain't that smart? So now we understand that squids use the laws of physics to camouflage. They have these uniformly spaced stacks of protein sheets that can selectively amplify certain color of light depending on the spacing between these sheets. And not only that, they can also change the spacing so that the same cell containing the same protein sheet can change from bright yellow to deep blue in a matter of seconds, all because the spacing between these protein sheets are changing instantaneously. Well, but I have to say, we do understand the science of how squid camouflage works, but we still don't know how the squid mind actually works. We know they manipulate light, use the laws of physics to camouflage, but as far as scientists know, squids are actually colorblind. So how they identify what they are doing still remains a big mystery, a big puzzle. They have their own language in which they communicate that still remains a big Morse code that we have not been able to figure out. And that brings me to our conclusion. Today, we actually told you the stories of 3 magnificent creatures that have evolved their own unique skill sets to actually adapt to their own environment. It's quite interesting to note that how they have left us these tiny puzzles to sort of figure out what is going on, how evolution actually helped them to sort of thrive in their own environment. And more importantly, it is remarkable yet humbling to think of us humans or Homo sapiens as yet another species who are going to leave our own footprint behind for a coming generation of species to figure out what we have done, how we thrived in our own environment. And of course, while each one of us would argue our own species is the best, it is up to you to decide which one is, and actually perhaps pick your own another entirely different species and tell us why you think your species is the best. And thank you. We'll be taking questions now.
So thank you to all of our speakers. I do see some questions that were dropped in the chat as we were talking. So if no one else has a question that they want to just shout out, I can go ahead and read some of those off. Okay. So from Gloria, She asks, they found mastodon bodies in Farmington, Connecticut. Are those relatives of the mammoth? So Tayah, I believe this question's for you.
Yeah, I put my answer in the chat, but yes, they
Oh,
are
perfect.
relatives. They actually broke off from the elephant lineage much earlier than mammoths, but they actually had some overlap in when the species were around. So that's kind of interesting. Thanks for the question.
Awesome. Okay, so there was an answer from Isa about Gloria's next question. I'll just read it for everyone. So the question was, is it true that noise pollution might be causing large whale beachings? And Isa says, yes, I believe this is true. In addition to being painful and frightening to whales, noise pollution limits whales' ability to navigate and feed, which would lead to an increased risk of beaching. And Ron has a question for Tayah. Why do we want to bring old species back?
Yeah, Ron, this question really opens the can of worms to this presentation, where there's obviously a lot of different answers here. You know, just the fact that we could, I feel like seems to be the major driver, but there's also the money aspect about it, the fame, the power. But at least in my eyes, I think the scientific advancement part of it is really exciting, but I definitely think the ethics and reasons behind it are something that the people involved should be considering before they go forward. But yeah, a great question and definitely something to chew on, before you go to bed tonight.
All right. Gloria has a question. If you put a squid in a tank and not in the ocean, do they still change colors? Anjana, what do you think?
Yes, they do. And in fact, the squid does not even have to be alive to actually make them camouflaged. So scientists have actually taken the mantle out of the squid and put it separately in a seawater base, and then sort of gave it, put some electrical rods up in there, and then sort of gave it a bunch of pulses in there, and they were able to see iridescence. So scientists still have to figure out how it exactly works, but we know that they can be done externally, even without the squid's command.
That's amazing. That's, I guess, makes me think of an analogy. It's like if you took a piece of skin off of a human and it wasn't attached to the body anymore, we would still see the same kind of biological changes or activities. Yeah, so that's
yeah, and they're colorblind, so maybe there is more information here. Like, they don't know that they are actually changing color. It could also be as easy as us breathing. We don't realize we are breathing. They don't realize they are changing color. If they don't even have to send a command all by themselves to change color, it could be, I don't know, quite interesting.